So now when we come to the process of DNA replication, we have initially there is the initiation step. Initiation step, in the initiation step there are the DNA A, DNA B, DNA C, 3 different proteins, and the primase and SSB and DNA polymerase. These are the other factors that is binding to the DNA during this initiation step. So we will see it one by one. The prokaryotic DNA replication during this process, the enzymes for the DNA replication are contained within a complex known as replisome, and this replisome actually consists of 2 parts. The replisome consists of a primosome. Primosome is with primase and helicase, or the primase and helicase enzymes forms a primosome, and 2 different or 2 units of the DNA polymerase 3 is also a part of the replisome. So in the replisome we have the primase enzyme and the helicase enzyme, and 2 different 2 units of the DNA polymerase 3 which is joined together, and this entire structure with the primase, helicase and 2 units of the DNA polymerase 3 constitutes the replisome, and the primase and helicase are known as a primosome. The replication fork moves in one direction and synthesizing both the strands simultaneously. That we need to understand. Previously, in the previous diagram that I shown, also the DNA replication is actually represented in a single direction with both the leading and lagging strands were synthesized on 2 different directions. But now we have to understand that DNA replication fork moves in only one direction and both the strands were synthesized simultaneously. And I will explain the concept. And in the previous picture, for example, in this one, the DNA replication moves in one direction, or the fork moves in one direction. But you know that always the DNA strand is copied from 5' to 3' direction. So this is the leading strand. This can be copied in the direction of movement of the unwinding or movement of the fork, so that this strand can continuously go on synthesizing during the unwinding. Whereas the second strand, the polymerase needs to start from here and copy from to here, and another strand needs to be copied from here to here. So the direction of copying of the lagging strand is just reverse. And now we are saying that we have a single unit of the replisome in which the primase, helicase and the polymerase 3, which will move only in one direction, that is in the direction of the replication. And during the process, it will copy the lagging strand also. It will not move forward and backward, to and fro. It will not do that for the replication. Usually, if you are representing a diagram like this, it is supposed to copy the DNA. Polymerase will move, or the replisome unit will move to this direction, and then it will return on the other strand to copy this one, and then it will move again here and move on the direction of the fork. So it will not move in both the directions. Now the fact is that the replication fork moves only in one direction, and during that process it will copy the lagging strand also using a special loop formation process that we will discuss. Anyway, for copying the lagging strand, the DNA polymerase will not move forward, or the DNA polymerase or the complex, replisome complex will not move it will not move backward direction for copying this one. And the DNA polymerase complex, that is the replisome complex, moves in one direction, and during that one direction or unidirectional movement, both the strands will be copied simultaneously. We will see how it is done. Now, come to the initiation again. In the initiation step, here we have the ORI-C of the E. coli. The E. coli ORI-C is a special sequence of about 245 base pairs, in which we have 13 base pair repeats. These 13 base pair repeat is a consensus sequences shown here: GA, TC, TN, TT, N, TT, TT. So this is a consensus sequence in which we have 13 base pair consensus sequence. So these 13 base pairs were repeated 3 times. So this is a tandem array of 3 repeated consensus sequences. This one. And there are 4 different 9 base pair sequences. And these are the consensus 9 base pair sequence: TT, AT, CC, ACA. This is the 4 or 5. In some cases, it is said as a 5 repeats of this consensus, so 9 mer sequence. So 13 MER, 13 mer sequence and 9 mer sequences were present in these 245 base pair origin of replication in E. coli. These 13 base pair sequence and the 9 mer sequence and 13 mer sequences were the actual initiation points or the unwinding region of the DNA replication in prokaryotes. Here the supercoiled DNA is shown. Here we have the ORI-C with about 245 base pair length, and the 4 base pair 9 base pair sequence and the 13 base pair repeats is there. Now the DNA A protein, the first protein or the first protein that is interacting with the DNA, which will join with the ATP, and then it binds to these 9 mer sequences, this region. It forms a loop, and that binding causes a distortion or a structural change in the origin DNA or the DNA strands in that origin region. So this structural change, which is caused by the binding of these DNA A protein ATP complex to the DNA, starts the unwinding of the or further structural changes and the unwinding of the DNA, which is required for a DNA initiation or replication. Now, there are other HU proteins and ATP, which is binding here, the DNA B and C, and with ATP it is binding here. And finally, this DNA B is the helicase enzyme. That DNA B is actually bound here, which produces the replication fork, initial part of the replication fork. So I will show you in detail in the next slides. So this is the overall initiation process. Here in this representation, this is the DNA A protein that is binding to the 9 mer sequence, which causes a structural change in the remaining 13 mer sequence, and there is a localized unwinding in that region. And the DNA helicase, which is known as DNA B, and the helicase loader DNA C, both of them will bind to these localized unwound region caused by the structural changes of these DNA A protein binding. And the DNA helicase loader DNA C will load the DNA helicase B protein to that template DNAs, and this creates the initiation of the, or this is the initiation of the eukaryotic replication process. Now, this is another diagram in which the DNA B protein is actually represented here, or the helicase is represented here, which starts unwinding of the DNA. And all these process requires ATP. Normally, we will discuss later. Anyway, this is the single strand binding proteins, and the single strand binding proteins will bind to the single stranded region. And another interesting fact is that once the SSB or single strand binding protein binds, then it promotes the binding of the other DNA polymerases into the single stranded regions. So wherever the single stranded DNA is present, the single strand binding proteins will bind to that region, it prevents the reassociation of or rehybridization or the hybridization of the DNA, and also it protects the DNA from damage, because the single-stranded DNA may be easy to be broken and susceptible to mutations or some other damages, but that is actually prevented by the binding of the single-strand binding proteins, which keep the single strands apart. So this is the previous presentation in which the RNA primer, or which is created by the primase enzyme, will bind, and so the helicase is bound, RNA primase is bound, and this RNA primase and helicase will produce the primosome, and the primosome creates an RNA primer. Then the DNA polymerase 3 is bound. Once the DNA polymerase 3 is bound, then the entire complex of the primosome and DNA polymerase 3, this constitutes the replisome, which moves in, the one replisome will move in one direction and the other one will move in other direction. So the replication in eukaryotic systems, where we have the two replication forks will be there, both move in both the directions, and which will finally meet at the termination site. So this replication or replisome, which contains the DNA polymerase 3 and helicase and primase, all these things will be the binding of all these complex, or the replisome complex formation is the initiation step of the prokaryotic replication. So the events during the initiation is actually mentioned here. DNA A ATP binds to the DNA at 9-mer sequence. The DNA A denatures or melts the DNA at 13-mer sequence, and the DNA B DNA C complex is loaded at the melted DNA. Then DNA B helicase and DNA C helicase loader, which the helicase loader will load the DNA B to that template DNA. Then with the aid of ATP, helicase unwinds the DNA. Then single strand binding proteins or single strands are occupied by the single strand binding proteins. Then primase forms a short RNA primer. Then finally DNA polymerase 3 is loaded and completing the formation of the replisome complex. So this is the different events that is occurring during the initiation stage. So once the strands are separated, then the strands continue to unwind. That continued unwinding of the DNA, that is done by the DNA gyrase enzyme. This DNA gyrase relieves the tension. The unwinding is actually done by the helicase, but the unwinding of the helicase creates tension in the DNA due to the supercoiling effects. And that supercoiling effects, which is or the tension that is created in the DNA, will be relieved by the DNA gyrase enzyme. It cuts nicks in both the strands of the DNA, allowing them to swirl around one another and then resealing the cut ends. Similar enzymes performed a task in eukaryotes. So there are topoisomerase enzymes which is doing the same function, isomerase 1 and 2 in case of eukaryotic systems also. Here the DNA gyrase enzyme in E. coli is the enzyme that relieves the tension of the supercoiling tension in the case of a prokaryotic DNA replication. And this is something like we can imagine a rope in which different threads are intertwined, and you can try to pull apart two strands of the thread of the rope, and the localized unwinding of that rope or two strands of the rope will create further tension or supercoiling effects on the other regions of the rope, and that can be relieved by cutting one or two strands so that the cut ends will swirl around or around the other strand, so that the tension will be relieved and the entire strand will be relaxed. So a simple model can be created using a rope in which you can try to pull apart the strands, then see the tension on the other ends or the region, remaining regions of the rope, and try to make a cut on that region so that it will automatically try to reverse by swirling around or turning around on the strands will turn around on one another, releasing the tension. And in the case of DNA, something similar happens when the DNA gyrase enzyme cuts the strands and releases the tension. Then finally, the cut strands will be resealed and joined together by the ligase enzymes, and that tension is relieved finally. Now, in the case of E. coli replication fork, these are the different enzymes that is interacting in the replication fork. I'm just listing that one. That is a SSB or single strand binding proteins, DNA B protein, that is a helicase, primase, that is a DNA G protein which creates the primer, DNA polymerase 3 which is used for polymerization, the DNA polymerase 1 which is used for removal of the primer and repair, and or the filling of the gaps. Then ligase, that is used for ligation or joining or sealing of the strands. DNA gyrase, which is a topoisomerase DNA topoisomerase class 2 enzyme gyrase, which cut both the strands of the DNA and releases the superfile. And when we discuss about the DNA polymerase topoisomerase one category, the topoisomerase 1 cut one of the strands and try to release the tension. And in case of topoisomerase 2, both the strands will be cut and the tension will be released. So DNA gyrase is a class topoisomerase 2 type enzyme that is used for releasing the tension caused by the superfiling. So these are the different proteins that is interacting at the DNA replication fork. Now this is the diagram. Previously, most of the at least at UG level, maybe you are discussing or you are using this diagram in which both the strands were represented, the leading strand and lagging strand. In the lagging strand, the fragments were synthesized in this direction, and leading strand, it is continuously synthesized, and the DNA replication fork or replosome will move in this direction. As I mentioned earlier, there will be a single DNA replosome which will move in this direction. So the lagging strand, how it is synthesized in the opposite direction, because the DNA polymerase is moving on in one direction, 3'5' direction of the leading strand continuously without moving back. So during that process, the lagging strand is also synthesized in the opposite direction, which requires a movement of the DNA polymerase in this direction. So how it is done simultaneously? This is a basic picture of DNA polymerase that may be used in some school curriculum or UG syllabus, but in the PG syllabus or when you are understanding the DNA replication at a master's level, you have to understand, you have to use this picture, not this one. This is a basic one which doesn't explain much of the facts. So here is the DNA replication fork where we have the helicase and the dimeric polymerase. So the helicase and primase will be here, which will move forward. Now this is the dimeric polymerase enzyme, which will one of the units will synthesize the leading strand, and the lagging strand will be synthesized by the second unit of the dimeric polymerase. And this DNA will produce a loop, and the primer will be synthesized here, and this loop will rearrange and it will be synthesized in the same direction. And I will show you a video how it is done. So by forming a loop and creating or reorienting the lagging strand in the same direction as that of the leading strand, by arranging these lagging strand in the same direction as that of the leading strand, the DNA replication moves in the same direction without the polymerase moving forward. Moving backward. So that's the actual concept. So this is the picture that may now onwards I am telling you that you can't use this picture. Always use this one with the loop. Now this is another one. if you are explaining DNA replication in the form of an essay, it's better to use this picture. Always mark all the items from one end. For example, from where will you start? You will start from this point. Here is the parent DNA. This is the DNA gyrase enzyme, which releases the tension, or the topoisomerase, which releases the tension accumulated due to unwinding in this region. And this is the helicase enzyme, which goes on unwinding the DNA strand. And these are the single strand binding proteins which keep the DNA strands single stranded and also protected. Now this is the polymerase enzyme with all the different subunits, and this is the clamp, sliding clamp, which keep the strands, the polymerase sticking onto the strands just like the cable car without falling off. This is the beta clamp, and these are the polymerases. And now these are the two subunits of the polymerase 3. Then this is the clamp loader protein. Then this is the lagging strand, and this is the primase enzyme which creates the primer. And by rearranging in this direction, the primer will be available here, and the polymerase can synthesize this strand in this direction. And starting from this primer, the polymerase starts synthesis or proceeds with synthesis in this direction. So the polymerase can move simply on single direction of the replication fork, and by rearranging or by forming a loop and rearranging the orientation of the DNA in the template strand, in the lagging strand, both the strands of the DNA is synthesized in a single step by this DNA polymerase enzyme. So this is it. Now we have the Okazaki fragments, which is present here with the primer and the Okazaki fragments, and the DNA polymerase 1, which is the repair enzyme, which will remove the primer and fill the gap. Finally, the ligase enzyme will seal the nick, or it will create a phosphodiester bond in the backbone at this point, so that both the strands will be sealed or joined together by the ligase enzyme. So these are the different factors that interacting. So if you are drawing the picture and starting from one point and ending it here, the entire DNA replication process with all the interacting factors can be represented. So nothing will be missing if you are using this picture. So this is the polymerization step, that is at the 3' end of the DNA strand goes in the 5'3' direction, and that there will be a free 3' OH group onto which the incoming nucleotide, the phosphate group of the incoming nucleotide will be attached, so that the phosphodiester backbone will be extended or in the same direction. So this is the actual chemical reaction of the formation of the phosphodiester backbone. So this is another one. There is a DNA polymerase 3. DNA polymerase 3 moves the RNA primers from the leading strand and the fragments from the lagging strand and replaces them with the appropriate deoxyribonucleotides. So primers were removed and it is replaced with the deoxyribonucleotides, so that the RNA is replaced with the DNA in that region, leaving the nick. The DNA polymerase 3 cannot seal the nick or join these fragments together. It fills the gap, but the nick is not sealed by the DNA polymerase 1. Now the ligase enzyme joins the Okazaki fragments, or this nick or this cut is actually sealed by the ligase enzyme, and that is the role of the ligase. So this is a mechanism of the DNA ligase reaction, in which the ligase enzyme with an here it is represented here, and adenylation of the ligase is actually done by AMP or ATP is attached here by releasing of the PPI from ATP. The enzyme is attached with the adenine. So enzyme AMP is formed. This enzyme AMP is produced from adenine by releasing the PPI. Then this AMP enzyme complex interacts at this region, and here is the nick in which the phosphate and OH, 3-OH and 5-phosphate of the subsequent Okazaki fragment or remaining Okazaki fragment. So these are the two Okazaki fragments with OH group and phosphate group. So this is created by the DNA polymerase 1, and this is the previous OH group that is existing. So this needs to be joined together by the ligase enzyme. So the enzyme, ligase enzyme AMP complex will bind, and then it will there will be an electrostatic interaction between these phosphate groups, which transfers the electron to the incoming adenosine AMP complex, and the OH group is transferred to the phosphate here, so that phosphodiester backbone is complete at that point, and the ligase will then release the ribose, adenine, and a phosphate. This is released by displacement of these AMP and with a phosphate. This group is released. So this is the ligase reaction, and the net result is that the seal, nick or the cut of the different Okazaki fragments is actually sealed by a phosphodiester bond. So this is about the DNA ligase reaction. So now I will show you a video.
Fork, two DNA polymerases collaborate to copy the leading strand template and the lagging strand template DNA. In this picture, the DNA polymerase that produces the lagging has just finished an Okazaki fragment.
This is the clone, this is the dimer of the polymerase, this is the primase, this is the helicase. So helicase, primase, DNA polymerase. So the DNA helicase will starts unwinding. The primase will create primer. So it unwinds, the primer creates the primer, SSB is bound, a loop which is formed orienting the strand in the same direction as that of the lagging strand, and now starting from the primer, it creates the or it starts the synthesis of the next strand, and after some time it releases the strand, and by the time the next primer is synthesized, and it reorients and aligns the DNA strand in the same direction and continues with the synthesis of the next Okazaki fragment. So this diagram, this animation shows exactly how the DNA strand is synthesized simultaneously, or both the DNA strands, leading and lagging strands, will synthesize simultaneously using this loop formation.
In a replication fork.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters
this is
the line from bottom left.
Process of the DNA
The
replication.
blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands.
So the next step is the termination step. in which we have the strands which is starting from the separation of the strands starts with the origin. The strands will move on both directions. The replication fork will move on both directions. There will be a termination site or ter site, and at the ter site there will be a TUS protein which will be binding to the termination. TUS protein will bind to the termination site, and at that the DNA polymerase enzyme cannot move forward or pass through this termination site which is bound with the TUS protein. So at that point it will stop the polymerization. Finally, there will be concatenate. Concatenate means two crossed rings, just like two rings joined, just no, okay. One ring will be inside the other one, so that without cutting one ring, the other one cannot be released. So that they are concatenates, and these concatenates will be formed. So that interlinked circles will be there. That interlinked circle will be released by cutting one of the strands and joining it together. So that is known as a decatenation. Decatenation by topoisomerase 2, which cuts both the strands and releasing both the strands of one of the circles, so that it can be released and resealed, so that complete two circles of the daughter DNA is produced during the DNA replication process. So there is a ter site which is bound with the TUS protein, and at that point the DNA polymerase stops replication process, and both the strands will join at that end termination point, and completing the synthesis, and the interlinked circles of the circular DNA or the concatenates can be released by the activity of the topoisomerase, the decatenation activity of the topoisomerase, which cut one of the strands and separate both the circles, then seal them together or join together with the ligase enzyme. So that is about the termination. So this is another schematic representation in which starting from the origin, the strands will move on both directions, and there will be ter A, B, C, D, G, F, and there are a lot of different TUS sites are there, and which will be bound with and both this it will be oriented in both directions. So for example, this one will move to this point and it will be stopped here, and this circle can move, it can pass through here and it will be stopped here. So this complete two circles will be produced. Otherwise, this will stop here, this will stop here, and this region cannot be copied. But these two termination sequence sets were arranged in two directions. That is, a clockwise and counterclockwise trap will be there. So the clockwise fork will end here. The counterclockwise fork will end here. So that both the strands, including this region, will be copied. This will copy up to this one. This will copy up to this region. Both the circle will be completely copied. And for this fork cannot pass this region. This fork cannot pass this region. So both the circles will be copied, but the circles will be interlinked. That will be separated by the topoisomerase 2 activity. So this is another the entire replication factory concept that is represented with because the replication machinery is not moving around. In prokaryotic systems, it said that it is actually membrane bound, and DNA molecules will be passed through that one. And the DNA polymerase is actually a heavy molecule which is actually fixed with the membrane bound molecule will be there. The DNA will be attached and it will be moving. So this is another representation of the replication factory concept, in which the polymerase will be fixed and membrane bound, and both the strands will be copied by the replication factory. Okay, now the next topics that we will discuss later. I think it's over 1 hour, so I will stop it right here. Any doubts? Hello?
No, sir.
Hello? Any doubts? സംശയമുണ്ടോ?
No, sir. Clear ആണ്.
ക്ലിയർ ആണ്. അസൈൻമെന്റ് എല്ലാവരും അപ്ലോഡ് ചെയ്തില്ലേ? ചെയ്തു. ഓക്കെ, എന്നാൽ നമുക്ക് സ്റ്റോപ്പ് ചെയ്യാം. ഓക്കെ ബൈ.